Hi, my name is Birge, and this is my topic, electrical brain stimulation. Electrical brain stimulation is a current of electricity that flows into the brain through one or more electrodes that will stimulate a response and create activity in large muscle groups and even the brain itself. Now, this stimulation won't be done on humans because in a high school setting, not many people will be willing to let a teenager poke around in their brain, and we also just don't have the resources available to test on humans. Instead, we can use insects, or more specifically, the Cotinus nitida. The Cotinus nitida was chosen because of its easy access to the brain, as well as its basic and easy to observe behavioral patterns. Experimenting with the electrical brain stimulation is important because it not only increases knowledge in a very unknown field of study, but they can also help aid against the effects of partial paralysis and depression or anxiety. However, this comes with some risks. Because the brain will be electrically stimulated, meaning there will be electricity flowing through the brain, if there's too much electricity, it can lead to tissue damage and ultimately death. Now, the first possible experiment that I could do is just a basic stimulation of the Cotinus nitida to evoke muscle movement in its wings to make it fly, essentially controlling its flight. In this picture, you can see sites where the brain is stimulated. For this experiment, the Cotinus nitida will be stimulated in the optic lobes, located in the occipital cortex. Because the optic lobes process information that was collected from the eyes and transmits that information to muscle groups throughout the body. Other possible sites of stimulation include the motor cortex, which deals with basic motor function and general muscle movements. When the brain is stimulated in different areas, different results will appear. A comparison can be made to how Frankenstein was created, where electricity was used in the brain to spark activity in muscles, or in Frankenstein's case, a living being. The second experiment is called electroconvulsive therapy. Electroconvulsive therapy is where the brain is stimulated in hopes to rid patients of anxiety, depression, or bipolar disorder. Electroconvulsive therapy is used as a last resort for patients when other methods have not worked. Similar to the previous experiment, this must be conducted on invertebrates. This can be done by observing sleeping and eating patterns, aggression, and abnormal activities of insects or the Cotinus nitida. The areas of the brain to stimulate for this experiment include the limbic system, or specifically the amygdala, hippocampus, and dorsomedial thalamus on humans. For the Cotinus nitida, the frontal lobe and parts of the temporal lobe would also be stimulated. When these parts of the brain are stimulated, they can reverse any adverse effects of depression or anxiety on the subject. Again, this is very important because there is not a lot of data on this known field of study and can have life-saving benefits to patients. I hope you enjoyed, and here are the works cited.